It's a pleasure to be here at OIS and introduce for the first time our company and technology. Ocular melanoma is a rare and life-threatening disease. It starts with a small melanocytic lesion in the back of your eye, and patients who suffer this disease have very poor alternative treatment options today. Depending on where the tumor is located, either patients will have to fear enucleation, the removal of the eye, or plaque brachytherapy or other sorts of radioactivity that unfortunately will leave them blind in a period of 2 years. We believe that Aura will radically change this for this group of patients. And we have a novel targeted therapy that can be administered in an outpatient procedure with an intravitreal injection activated with an ophthalmic laser that can preserve vision and remove these tumors. We are very, very happy because this year we received the orphan drug designation by the FDA. And we will have clinical trials started, phase 2 started next year, very early. And we will have a very early readout. If we are successful, for this drug, we will have a very nice path towards commercial launch. As a rare disease, it will take a small number of patients, and if we demonstrate safety and efficacy, this will be the very first drug without competition for patients that have a huge unmet medical need. We're excited. We have a management team in place, and let me show you who's behind the company. Aura is a venture-backed company. We have enough capital to run all our clinical trials. We're very excited. We've put together an extremely experienced management team that has expertise in the rare disease field. And we've also put together a group of KOLs that are the top world opinion leaders in ocular melanoma. We have Dr. Carol Shields, Dr. Evan Gregudas at Mass Eye and Ear, and Dr. Arun Singh at the Cleveland Clinic. So let me tell you a little bit about our technology. Our technology was discovered at the National Cancer Institute by Dr. John Schiller. It's based on the capsid of the family of papillomaviruses. The discovery is that this capsid, this protein that forms the virus, has a very selective targeting to tumors. Not only ocular melanoma, but mostly all solid tumors share a molecule on the surface called heparan sulfate proteoglycan. It's a type of sugars that are modified and highly expressed in tumors. We've validated that this is actually present in all samples of ocular melanoma patients in paraffin-embedded samples of enucleated eyes. And this protein can be used in 2 settings. It can allow us to deliver DNA, as it would be for gene therapy, but it also allows us to deliver a huge amount of small molecules, much more so than antibodies, which can deliver 3 to 5. Here we're in the range of 300 to 500 molecules, up to 1,000, which you can imagine the potency of these molecules when they reach their target. So now let's focus about ocular melanoma. We are excited, and the reason we are excited is because there are a lot of patients that don't have any treatment option for this disease. And when you face that, everything becomes very meaningful. And it's very important because in all these diseases, in all of the phases, the unmet medical need is very big. It starts primary, confined within the eye. It's probably the tumor where we diagnose when it's 1 millimeter inside of a cavity and not yet metastatic. What an incredible opportunity to treat these patients and change their lives. Instead of that, because there's no drug therapy today, we wait until the tumor grows. And then, as we mentioned, we have the alternative options that you will see. So Aura is very focused on the primary stage, which must not be confused with the metastatic stage. This is what these patients face today. If you have ocular melanoma, you're 35 years old, you have this small lesion in the back of your eye will be removed, there will be a plaque inserted in the back of your eye filled up with iodine radioactive seeds, which will be placed for 7 days, irradiating your eye. And then the eye will be removed again to remove the plaque, and insert it. As you can imagine, the side effects that these patients go through after treatment are really terrible. They have glaucoma, retinopathy, dry eye, all of the ones that we're trying to correct with the previous presentations, they probably experienced them all. The tumor stops growing, but their vision, unfortunately, is lost. So this is how our drug works. As I mentioned, it has two components. It has the viral particle and the small molecule. So the viral particle binds the tumor very selectively, but it really doesn't create any damage. So when we inject these patients intravitreally, it basically is a prodrug. The drug accumulates in the tumor in the back of the eye, and then with a regular ophthalmic laser, the same laser that has already been approved for the use of wet AMD, and it's available in all of these ophthalmology services, is used to irradiate the tumor, and only the tumor. And upon irradiation, the small molecule activates, and it's extremely powerful. It creates a very rapid necrosis, and the cell dies. So we've developed an animal model. We've used 2 academic sites plus a CRO. The model is extremely relevant to what we see in the clinic, because in a rabbit, we can actually induce an ocular melanoma in the same location that it grows in patients. These tumors grow exactly the same way as a patient would develop an ocular melanoma, and we can treat them intravitreally and laser them as we would do in the clinic. So we've done this in multiple independent experiments. And what we see after 3 intravitreal injections and laser activation, when the tumors are small, they don't just stop growing, they disappear. There's no tumor left by histopathology after treatment. When the tumors are very big, those that would go to enucleation in a patient, we see overall necrosis. And hopefully those tumors in a longer time would also disappear. So that's the opportunity that we have for patients. What's our clinical strategy? We're going to start next year. As I said, we have 10 hospitals lined up, and it is a rare disease, so we want to make sure that we enroll rapidly. These are probably the best 10 hospitals in the United States. So the first study is a dose-ranging study. We're going to test 2 doses of AU-011. And hopefully what we will see is safety and dose response. Once we demonstrate safety and efficacy for these patients, these are small tumors that are growing and would be selected for plaque radioactivity, then we would go into a trial for registration of those tumors that are really, really small and not yet growing. We have checked our clinical development plan with the FDA in 2 pre-IND meetings. This is, as you can imagine, a very novel development. No one has ever developed a drug for ocular melanoma. And so we're very thankful that the FDA has provided a lot of advice to design this clinical regulatory path. So what else does Aura have? Of course, as I mentioned, our drug is not just for ocular melanoma because our viral particle is very selective to all tumors. However, we want to really provide the best treatment option for ocular cancers, starting with ocular melanoma. We can then treat retinoblastomas, conjunctival melanomas, conjunctival squamous cell carcinomas. There are so many that have nothing today. After that, our company will work to develop the technology systemically for head and neck cancer, bladder and prostate cancers, and we also have a very exciting development in immuno-oncology. So as a summary, we should all feel very excited when we treat a rare disease, especially in ocular melanoma, because it's a rare disease that has no drug therapies approved. So hopefully what we'll be able to demonstrate is that we have a targeted therapy that's very safe, that allows us to treat these patients in an outpatient setting, and that we can really change their lives for the longer term. Thank you very much.